It's a really great pleasure to have Olga Troyanskaya here today. Olga got her PhD in Stanford, and she's now a professor in Princeton and also a deputy director at Flatiron Institute of the Simons Foundation in New York. She won, obviously, many, many prizes, including the Overton Prize for Computational Biology. And she is just a world leader in computational genomics, and I'm just very excited to hear what you have to tell us today. Thank you.
Okay, I'll try this. I broke my arm in China, so I apologize that this will be creative. All right, it's a great pleasure to be here. I have lots of friends here in the computer science department and at the genome sciences department in the medical school, and so it's great to have all of you guys here. Just so I can sort of figure out, judge, can you guys raise your hand if you're mostly, like, you don't know much biology and you're mostly from, some computational field, but don't know much biology? Okay. Good. And then you're mostly biology but don't know much about computation? Okay. So I'll try to get both audiences as opposed to confusing both audiences. But please do ask, especially clarifying questions. If something is not making sense, there's probably other people in the audience to whom it's also not making sense. All right. So without much further ado, I want to, let's see if I can get this to work. I want to tell you about essentially the main problem that we think about, and I think by we, I mean not just my group, but really in general, a huge number of computational biologists, clinicians, population geneticists, etc. And that is of how, if we think about complex human traits, or more specifically, complex human diseases, by which I pretty much mean the vast majority of diseases you can think of. Here I have a child with autism in the top picture, and sort of a picture of a blood pressure monitor really symbolizing hypertension. But anything, kidney diseases, diabetes, cancers, right? All of those are complex, by which I mean there's many genes that are probably underlying your susceptibility to this disease, how this disease should be treated, etc. The fundamental question that we need to figure out, if we know that the disease has a genetic basis, which for pretty much all of these diseases, or the vast majority of these diseases, we actually do know this scientifically, is what is this genetic basis? What are genes and specific variations in the genome that are really underlying both your susceptibility to getting, for example, Alzheimer's, or a particular type of cancer, or maybe actually being resistant to getting Alzheimer's? And also, understanding if somebody does get a disease, what are the specific genetic characteristics in their genome that might help us diagnose and treat this disease better, right? So here there's this picture of binders, and the binders are the Human Genome Library at the Wellcome Collection in the UK, and it's literally the human genome, the over 3 billion letters of the human genome printed out. And each folder has a number on it, like 13. There's multiple binders there. 13, that's the 13th chromosome. So it's literally looks like this if you open it, right? And this is very informative. So we know some things about the human genome. We know some of those areas in blue are genes. We know some of the regulatory signals there in green that might tell us when genes are being turned on and off. But the vast majority of the genome, we really don't know how to interpret. So if you come to me with a genome and you say, I have this mutation here, or I have, Hank, the CS department chair over there, maybe has an A in this position outside of some gene, which is the vast, like 98% of the human genome is outside of genes, right? So he has an A there, I have a T there. That means absolutely nothing. Or it might mean that, Hank has straight hair and I have curly hair. Or it might be actually a critical thing to determining our predisposition for a particular disease or our ability to metabolize a particular drug. So how do we start studying this? And so I want to tell you about how my group thinks about it, and we think about it sort of through the common words that biologists talk about is the central dogma, and that is how DNA becomes RNA and protein, and then actually leads to what we call a phenotype, or really, disease, health, or color of your hair, basically some observation about, in our case, a human being. And the challenge here is, I'll just tell you one fundamental, beyond the disease, one fundamental puzzle. The genome is the same in every one of your cells, right? Every single one of your cells essentially has the same genome. And it started with two cells, but one, right? Like one single cell, really, out of two cells fusing. So how is it that your brain cells are so different than your liver cells? In fact, within the brain, there's huge differences in different cell types. So that means then we need to first try to understand how we decode the sequence of this genome. And that means that we need to figure out how does a single letter change in the genome affect how genes are turned on and off and regulated. Furthermore, we need to understand not only how does it regulate this basic molecular characteristic, but also what does it actually mean in terms of whether that single letter change is functional and whether it leads to disease, right? Because it could be functional but benign, right? Like the hair curliness factor that I talked about, right? It's interesting, but it's not so important, but for most of us at least. But, predisposition to Alzheimer's is very critical, right? So how can we predict that and understand that? Of course, now in biology, for the past 20 or so years, we've really been experiencing this incredible revolution where we are getting not just genomic sequencing, but a lot of data on cells that's sampling different biomolecules. So the vast majority of this data is really about genes being turned on and off and measuring how much each gene is being turned on and off in a whole genome scale, right? So for every gene in a cell. And being able to do it across many conditions. For example, many patients with Alzheimer's or breast cancer, et cetera, or in cells that are sitting in Petri dishes, right? Like watching a cell go from an embryonic stem cell to becoming a beating cardiomyocyte, a beating heart cell, for example. Then, of course, once that gene is expressed, once it's turned on and it makes a gene product which actually does something in the cell, those gene products don't function by themselves. They work together in very complex networks. So for computer science and engineers, the best analogy I can suggest is basically circuits, right? So just like in circuits, there's this complex connectivity between circuit components. In these biological networks, there's complex connectivity and interplay between different gene products. And that connectivity is actually different in different tissues and cell types, right? So it's different in your liver cells versus your brain cells. In fact, it's vastly different in different types of neurons in your brain. And this is not just, it's very interesting biologically in understanding fundamentally how we are made. But if you want to really think about enabling precision medicine, which I'm sure UW Hospital probably has advertisements just like every hospital in New York does, that they're going to treat your cancer and sequence your genome, right? If we want to actually be able to do it, we need to be able to understand how these networks change in different cell types, right? Why is it that some neurons die from Alzheimer's very early on in disease, and others that on molecular level, actually, on a high level that we've studied them so far, we the community have studied them so far, are very similar, are highly resistant. In fact, that they don't seem to have affected by the disease. The patient usually dies before those neurons are in any way affected. So if we can understand that, we can actually potentially understand really what's going on in Alzheimer's. So we need to be able to map these networks and pathways across tissues, and this is a very hard problem experimentally, of course, because most of us don't like giving brain biopsies continuously, for example, right? So this is kind of an extreme way to explain to computer scientists, but realistically, right, how do we study this in human? How do we study what do these different networks look like in different cell types in the brain? And that means that we have to solve this computationally. We either have to try to detect it from mixed samples that are mostly bulk tissue, and a lot of times from basically subjects who are dead, post-mortem subjects, mostly motorcycle accident victims. Don't drive motorcycles if you can. No, literally, if you look at those collections, it's all young men. But also, we need to start thinking about how we can use model organisms. So biology use mouse, worm, fly, yeast, right? These are called model organisms for a reason. While they're all, well, okay, at least mice are cute. I'll show you some worms. They're reasonably cute. My tray will be mad at me. Yeast are sort of cute. But the point is, these are not studied purely for their own sake when we think about them as model organisms, but because they fundamentally allow us to study mechanisms in a way that we couldn't possibly do in human, right? In mouse, we can actually cut out the brain of the mouse and get all of those different neurons in a perfect situation where it's, it goes into liquid nitrogen immediately. But of course, then there's a lot of computational questions of how relevant is it to human biology. So I'll talk about that as well. So can we use these maps to identify tissue-specific genes and really fuse the information that we get from model organisms together with data from population studies in human, which is, of course, much less resolved for those cell types for the reasons that I've talked about. So without much further ado, let me answer the first question, or at least tell you how we started answering the first question. And that is the question of trying to interpret the 98% of the genome that is non-coding, that is outside of the genes that, for a long time used to be called junk DNA. And that's the part of the genome that really encodes a lot of the regulation of how genes are basically turned on and off. And that's what really underlies this huge diversity of cell types that we have given the same genome. So, right, you all have the same genome in all of your cells, but it's which genes are turned on and off when that makes them, those cell types, different. So there's a question of mutations, right? Back to the question of disease mutations. And when we look at mutations within a gene, there's actually the genetic code, literally a table that you can get out of any genetics introductory textbook that has a translation of three-letter words, right? ATT, for example, and what particular amino acid it encodes in these protein machines, right? So you literally know the exact, or more or less, this is not a completely solved problem, but we have a pretty good understanding if this T changes to an A within a protein of where it affects things in the protein sequence, right? So we kind of know how it screws up the machine, right? That's going to be part of the circuit. There's still very interesting problems there, but at least we have some understanding. Now, what happens if we're outside of the genes, right? In this 98% of the genome that is non-coding. Now, not simply is this huge part of the genome, but also the vast majority of variants that have been implicated in disease from big studies of huge groups of people that are healthy or diseased, genome-wide association studies, over 99% of those variants are in this non-coding space. So what do we do with them? Well, so in reality, one of the biggest ways that they're going to affect things is how genes are turned on and off, and that's done through this complex structure of proteins and other biomolecules that is associated with DNA in the cell when it's packed, and it's called chromatin. So chromatin is a number of proteins that basically, when DNA is sitting in the cell, it's not just this one long letter sequence of ATCs and Gs, it's packed in this very complex structure, and there's many proteins that it's packed around. Those proteins can be modified to basically open up the DNA so that other protein machines can get in and turn these genes on. And that structure is really critical to this regulation. So what we need to do is we need to figure out this chromatin code so we can interpret non-coding variants. So Jan Zhou in my lab has developed a deep learning sequence model, and the goal of this model is exactly that we can take the genomic sequence and use the sequence model to interpret what this sequence corresponds to in chromatin organization. So just to give you a specific example of what this model does, and then I'll actually, the computer scientists don't fall asleep, I will show you what the model actually is in a second. I just want to show you what I'm talking about first in terms of practically what it means. That means if you give me a genome and you tell me, well, here, this letter changed from an A to a C, there's this red C here, then we can take our deep convolutional neural network model, so the network has already been trained, I'll tell you how in a second. It can predict the effect of this specific, change on a number of hundreds of different marks on these proteins that DNA wraps around, how open DNA is, and particular protein machines that specifically bind DNA to turn genes on and off, transcription factors. So these are really important proteins that turn other proteins on and off. And that means that if we had a healthy individual, for example, that had an A here, that was a sequence that encoded a message to one of those transcription factors to bind here, versus once this was changed to a C, then perhaps the transcription factor no longer binds and it falls off, and that may cause disease. So that's what the model actually does. And the way it works is it's a deep convolutional neural net. It has a convolution layer that basically scans for motifs. So we scan it in windows through DNA. The intuition for the pooling layer is that it does spatial scaling, because in DNA, remember, DNA is wrapped around stuff in the nucleus, so there's complex relationships between things that are not linearly adjacent to each other, so you want to be able to capture that. And then finally, there's a convolution layer that tries to capture even higher order sequence features further away from each other. So really, the hierarchical structure allows you to capture this context information of each feature, of each sequence. The multi-layer nonlinear transformation allows us to encode the interaction of different sequence features, because we know from biology, and we know from the way the model now works, that these features do interact, right? So different molecular biomolecules that I've talked about do interact with each other quite extensively. And the multitask prediction at the top allows us to share all input, because there's multiple cell types for which we have this information, and cell types do share some characteristics, but they're different in other ways, right? So different subtypes of neurons share some signals, but are not all identical, and so that means that cell types for which there's less data can actually benefit from borrowing relevant information from cell types where there's more data. This actually works quite well. I'm just going to show one holdout evaluation. I'm happy to show you way many more slides of evaluations or tell you afterwards in questions, but basically this is just literally hiding a whole chromosome out of the whole setup, right? So we pretend like there's no chromosome 18, I believe it's 18, but doesn't matter, in the genome, and we never show it to the model, and once we train the model, we apply it to that chromosome. And it does quite well. This is across over 100 different cell types, training on the ENCODE epigenetic and roadmap data for the biologists in the audience. So this is basically large NIH-funded studies that try to measure these chromatin profiles, right? So the model in training never sees mutational data. This is key to this question. Because, of course, the biggest challenge is that we're never going to see the most interesting mutations. This is because evolution is a great thing for us not getting as many diseases, right? Because basically mutations that are disease-causing are selected against quite heavily. And the non-coding space is so vast that it's unlikely you'll see the same mutation twice, also for that reason. So even if we sequenced every person on Earth and had clinical information on them, we're still not going to be able to learn this in a supervised setup. So basically in an example, like trying to match mutation A to all the people who have it and have the same clinical phenotype. So that's why we really have to be able to learn really the deep sequence features that underlie these signals and then be able to predict what happens. So now you can take a Neanderthal genome and predict what it would look like in terms of chromatin, right? Because really you're learning how does the sequence encode this organization. So what can we use it for? We are currently using it to look at autism. And autism, I'm sure you've all heard about, is highly prevalent. I think some estimates 1 in 64 kids in the US is affected. This is a study that's in very close collaboration. So on our side, from Jan Zhou, again, with Chandra Tesfail, who's an amazing experimental research scientist in my lab, and with Chris Park, who is now a research scientist in my lab, but was at the time a postdoc with Bob Darnell's group at the Rockefeller University. So here's the setup of this study. This is the Simon Simplex Autism Collection. They have over, I believe, 2,500 families that have this quad design. That means that there's mother, father, and 2 children sequenced in every one of those families, whole genome sequencing. So this is a pretty unique resource. And in each family, there is a child, what is called proben, a child with autism, and a sibling that doesn't have autism. So we have genome sequences of all of them. And in autism, as well as in many other diseases, there's been clear evidence of what is called de novo mutations being important. And what de novo just really means is a fancy word for the fact that you sequence the mother or the father, they don't have the mutation, and the child with autism or the sibling does, right? So de novo just means it didn't come from your parents, it appeared just in you. So we have over 7,000 whole genomes in these families, and the key question we're trying to ask is, and this question actually is not just a big challenge for autism, it's for any complex human disease. There hasn't been any proof that de novo non-coding mutations actually have real relevance to human disease. While there's many examples, single examples that we've seen, it's actually a pretty critical question in genetics whether there is real statistical proof of this. And people have been actually looking at this data and not finding statistical proof. And the way you would prove it is you would literally count the number of mutations, de novo mutations in the autistic kids, count the number in their unaffected siblings, and say, look, the autistic kids have significantly more mutations than their siblings, so therefore, there's clear non-coding signal. That doesn't work. Because of multiple hypothesis testing, you basically can't, there's no significance that people have shown. Well, people have shown that there's no significance. There's literally an Nature Genetics paper just saying that you can't do it with less than, I think, 8,000 families. Now, what we decided to do is, instead of looking at how many mutations, we asked how bad of mutations, right? Because now we have this model that can predict the impact of mutations at the biochemical level, and we can just put it in a classifier and say, well, this biochemical impact, how similar to a disease mutation does it look? And that gives us a disease relevance score, purely based on this biochemical impact. And all of a sudden, we have a complete statistical significance for all variants, right? There's no genome-wide hypothesis issue. We're literally looking at every single de novo variant, and we have a significant difference between siblings and probands. I'm sure it's a little bright, so you can't see it. You just have to trust me. Here's the p-values are visible. And it gets even more significant if you look at variants that are close to genes, and even more significant if you look at genes that are more likely to be disease-associated. So these are genes that are evolutionarily conserved. And we see that independently at both the transcriptional level, the DNA chromatin model level, which I talked about, and we also have an independent model that looks at post-processing, so RNA processing level, which is a different level of basically making a gene into a protein, and we see the same exact signal there. So we've actually also tested about 34, I think, of those variants and got 94% confirmation. Of course, this is not proof that these variants are autism variants. That's a very challenging thing to prove. But remember, they did come from these families with autism. But this is proving that these variants do drive difference in transcription. So this is literally taking what this variant looks like in a sibling versus the child with autism and saying that and looking in a cell in a Petri dish, that there is more RNA made, right? So the genes that turn up more or less significantly based on just changing that one letter in the exact context that you see in the sibling versus the child with autism. So 94% of those that we predicted and looked at, we confirmed. Now, can we go further? Can we actually now not just predict the biochemical effect, but actually predict gene expression? So can we, with my Neanderthal genome example, can we literally take the Neanderthal genome and read off how much of each gene it would have based on purely the genomic sequence? Or take a patient genome and say how a particular mutation in the non-coding region would affect expression on any gene in the genome. So we've addressed this problem here for 218 tissues and cell types that have been looked at in the GTEx compendium by, again, another NIH study. The way we do it is we actually build on a somewhat more expanded model of the deep learning framework I talked about earlier. So the first part of the framework is exactly what I told you about before. We basically predict the biochemical setup, right? So for those of you who are experimentalists, we're literally predicting, or computational people who work with biology, we're predicting in silico chromatin reads, right? So in silico ChIP-seq, completely based on purely sequence. For those of you who are computationalists, remember I just told you about all of those biochemical features. That's exactly what we're doing, but we're focusing it right around the start of the gene, because we know that's where a lot of the regulation happens. So then once we have that, we can actually train so we have to do a spatial transformation, that's important for basically just reducing the dimensionality of the problem and also focusing it even more on that area in the start of the gene. And then we can actually train just a simple regularized linear model with the expression data, right? So the model knew nothing, again, about mutations and never trained on matched genome to expression data. We're just training the expression data based on the predicted ChIP-seq data, right? And now we can basically, purely based on sequence, predict the expression of each gene in the genome in every one of those 218 tissues, as well as predict the effect of expression on the expression of every one of those genes in the genome in 218 tissues. And that would allow us to basically do in silico mutagenesis to see what effect every possible mutation around the start of the gene would have. So we looked at 140 million mutations, which is 1,000 base pairs around the start of each gene in the genome, which allows us now to look at, remember I told you the vast majority of those variants that were implicated in disease, but we weren't sure are disease associated, lie in these non-coding regions. Well, now we can look at each one of those variants and say, well, does it look like it should change gene expression? If it does, it looks like it's much more likely to cause disease. And also now, what is a bigger, really completely open challenge in genetics, we can now try to look at truly de novo variants, right? So variants that are so rare that you can never pick them up in population genetic studies, right? Because if we have a variant that seems to have a very strong predicted effect on gene expression, and that gene is a gene that's implicated in a particular disease or looks like it affects a pathway that's very relevant to disease, that's a potentially very good candidate for a de novo variant. Does it actually work? Well, first of all, we can just, again, do our chromosome holdout trick, hold it out from the entire process, and see if we can predict expression of those genes. And the expression correlation of this predicted expression to the true expression across those 218 tissues across every gene in the gene, well, every gene on that holdout chromosome, is over 0.8. So that's actually very impressive, but you shouldn't view it as perfect at all. Correlation of, if you take two patients, for example, in a study, their expression correlation would be probably over 0.94, right? So this is very good, but if I told you that I got 0.94 purely based on what I just told you, you should know I'm lying because I'm not incorporating anything about the environment, right? So I shouldn't be able to get perfect correlation. But it's quite good, and we actually have carefully looked. It's not just global correlation, it's actually tissue-specific as well. And we can actually use this to very accurately predict what's called expression quantitative trait loci, so basically variants, mutations that people have in expression studies identified as regulating expression. So you can see that when Expecto, our tool, predicts that a variant has a high impact of expression, it's nearly perfect across these 4 very large studies that identify DQTLs. Now, I promised you that now we can actually identify potentially which of the variants that have been associated with disease are really associated with disease. So we've looked at 8 different diseases, 8 different variants across immune diseases. I'm just going to show you this one graph and then just click through just to show you that it works, so experimentally. So what we did is we actually looked, for example, in this case, the genome-wide association studies, this was across 3 different diseases. They were all looking at variants related to this one gene, and they identified all of these different variants. So, in their papers, they said, this is the one. And it was each one of those 5 crosses, I guess, was the one in the different studies. Expecto predicted that none of them change expression at all, except for this one that none of the GWAS studies actually pointed out to. And if we look experimentally, whoops, sorry, I'm really apologize if I'm like shining the laser in your eyes. Ed did predict this, I'm sorry. All right, it's all Ed's fault. All right, so if we look, our predicted SNP, causal SNP, we'll look at that again in this, Petri dish cell, so it's not a perfect validation, but it's real experimental validation. We just ask, if we make the exact change that we are predicting, does it actually drive a change in expression of this gene? And yes, our variant does, and none of the other variants drives it to change significantly. And that's the same story that we see across these other variants as well. So that was true for all of the ones. We just tested all the top ones for our immune predictions, and they were all correct in this case. Now you can get to I'm a big believer just for the computational, especially computational biology students and postdocs in the audience, I think it's really important that we make our tools available to biologists, both through collaborating with biologists, but also through really enabling them themselves to use them. So we have this tool called HumanBase. It's basically sort of our, not at all in algorithmic sense, but Google for data-driven biology. It has access to all of those Expecto predictions, and you can browse them. And this is really work of the HumanBase team led by Aaron Wong. We also believe in making open source library to enable other computational people to get into this. So we are hopefully in the final stages of getting this manuscript through review for a deep learning library, specifically for sequences in genomics. So we would love for you guys to use them, and it's completely open source as well. All right. So now remember I told you that once the genes are expressed, so this is great, we can predict expression, but once they're expressed, they act in these complex networks, and these networks are very hard to really map in specific cell types. So how can we map them? And here I really want to tell you about the work of Vicky Yao, who is a brilliant postdoc in my group who recently graduated and is actually applying to your department, to CS department here. This was recently published in Nature Biotechnology. So she developed a method for making semi-supervised cross-organism networks that really allow us to build these networks with very little prior data, prior knowledge, which is, of course, a huge challenge here. And then we have prior work on non-semi-supervised simple Bayesian methods for this from Casey Green, Aaron, and Arjun from 2015 in humans. So how do we do this? So remember, we have basically, this is a kitchen sink method. We have a huge number, thousands and thousands of data sets, mostly of gene expression data. So this is for every gene in the genome, how much of it is expressed across huge variety of very diverse conditions. We have a little bit of sequence data and some data on how proteins physically bind each other. And then what we want to be able to say is how do circuits look like in different cell types, even for cell types for which we have not a single data set, right? Or cell types for which we have very little data, right? So essentially, how can we extract these data? We want to use a hook of gold standards, of examples to be able to extract the relevant signal from these messy data collections. So we're going to use a machine learning classifier, which is, in the basic case, a naive Bayes classifier in this case. And the naive Bayes classifier is basically the first thing it will do is do weighted voting. So I apologize for the basic explanation for those of you who work with Bayesian networks, but I find it very useful to think about it as essentially giving how good is a data set in reflecting the examples of genes that we know interact in a specific tissue, right? So what we ideally want is basically pieces of circuits that we know are happening in a particular neuron on the brain, and we see how good is each data set in reflecting those pieces of circuits. And then each data set gets basically a weight for a vote. It would be great if our elections were done that way. If you only got to vote and your weight of voting was proportional to how much you actually read up or thought about the problems. All right. Maybe it's controversial. All right. And we can use this, right? Once we have those weights, we can use those weights to essentially figure out what the probability of a true functional relationship for two genes is based on what their observation is in each data set, right? How, for example, they're correlated in each gene expression data set and factoring in the weight of that data set, right? Because some data sets in this collection are just crap. I'm sorry, this is a professional word. Some of them are wonderful, but they're completely irrelevant to for example, that neuron in the brain, right? But they might be really amazing to something else. And the problem is it's very hard to assess without doing it computationally, right? Because honestly, even some of those data sets that are garbage might actually be your gold mine if they have the relevant, they might have actually done not the experiment they thought they were doing, but they might have perturbed the processes you care about for that prediction, right? So it really has to be done computationally. Now, the semi-supervised part comes from the fact that, remember I told you that it's very hard to look at those circuits, and for most of those, or many of those cell types that we really care about, we actually don't have any examples of circuits, or very few examples of circuits that we can actually use as our gold standard. So essentially what Vicky does is that she came up with an idea that we can, instead of using true positives and true negatives, she's going to weigh the positives and the negatives that are predicted by a prediction method based on these data, right? So basically she is doing it in a supervised way and adjusting her probabilities based on how likely that prediction was to be a positive relationship or a negative relationship. So she's basically predicting the pieces of circuits that she's going to use to weigh the data sets. And then using that, those confidence scores to be able to make confident predictions. So that means that once she repeats this for all gene pairs, she'll get a network, and she can do these networks for all the different cell types and tissues. And it turns out that these networks are basically, nearly in all cases, across all of the different tissue cell types, much better at predicting tissue-specific circuits than networks that are not trained in a tissue-specific way. And furthermore, in nearly all cases but a couple, it's much better to do the semi-supervised training than the standard supervised training, right? So you really do need to be able to do both of those things to do well. And you can get to the human networks through our human-based resource that I've already mentioned, and biologists, well, or computational biologists, or computer scientists, if you have free time, can look at any genes that you're interested in. And in this case, we're looking at this important regulator in inflammation, IL-1 beta, and our collaborators have actually tested these predictions, and they are correct. Now, going further, remember I told you that it's very hard to study many things in humans for both technical reasons and largely for just ethical reasons, right? I mean, it's very hard to do experiments, well, you know, any sort of invasive experiments we don't do in humans. Let's just put it this way, simpler. So how do we study these mechanisms, especially in cell types of the brain that we literally can't get to at all, right? And what Vicky has addressed in this study is really thinking about the question of, can we use data from other model organisms, so in this case mice, where we can get at these cell types, and try to answer this question. So this is really addressing that question I've raised earlier as an example, the question of selective neuronal vulnerability in Alzheimer's, the fact that some neurons are very vulnerable, they're the first ones to die in Alzheimer's disease, and others that are very similar to them are completely resistant. If we can understand this, this is a key potentially to understanding really what's happening in Alzheimer's. Alzheimer's research has been, you know, a huge amount of research is being done, a huge amount of money is being spent, and it's been incredibly productive, but we still basically have one sort of gene that we know is involved, and it's a very challenging question. So here we have our collaborators at Rockefeller who can create mice for 7 different cell types, 2 cell types that are specifically vulnerable to Alzheimer's and others that are specifically resistant. And Vicky used this information to actually computationally map relevant mouse cell types to human and show on a molecular level that these cell types do actually map to the relevant human brain regions, right? So this is the fundamental question you need to answer in any of these studies. Can we actually use the mouse brain to study human brain? And yes, on the molecular level, there is real conservation here. So then she actually can use human gene expression data collection, right? The collection I just talked about. But mouse cell type specific expression, so what we know about those cell types that are vulnerable or resistant to Alzheimer's in mouse, where we can't get at this data at human at all, to make human networks, right? So the hook we're using is the mouse cell type specific expression data, but human data collection to make human networks for these vulnerable cell types. And then she developed this method for being able to use human quantitative genetic studies, right? So very large studies where people looked at patients with Alzheimer's disease and healthy people and basically identified genes, so essentially ordered all genes by p-value of how likely they are on the genetic basis to have some association with Alzheimer's, right? And most of them, because of the issues with statistics and multiple hypothesis correction, there's very few real significant associations, but you do get a p-value for everything. So what we can do is we can say, well, there still should be signal in the top of this list, even if they're just nominally significant, right? So not multiple hypothesis corrected. We can take those guys, throw them onto the brain network, and then Vicky takes the negatives and probabilistically subsamples them to provide robustness. And then we put the network, the brain network, into a support vector machine classifier. So essentially her idea is that we can identify network patterns that are relevant to Alzheimer's based on the disease signal that is not apparent but hidden in these GWAS studies, right? Because of statistical significance, you can't actually just pull it out. But based on the network pattern differences, we can pull out those signals that are beyond the statistical significance threshold but real. And that allowed us to actually, and our collaborators, of course, to identify specifically 4 modules of genes that seem to be involved in Alzheimer's in human that are specific to neuronal vulnerability, where one of them is this module C, this axonal module, is really one that underlies, the only one that really underlies the selective neuronal vulnerability in Alzheimer's. And it's actually very interesting that actually it's very much linked to tau, which is a protein that has been implicated in Alzheimer's already. And I'm happy to tell you more about it, but I want to move on to just to leave you with the key point that it actually, they've verified some of our predictions experimentally, and it looks extremely interesting and involves selective exon exclusion, et cetera. All right. So can we actually go further? Can we systematically integrate the model organism information with the human quantitative genetic studies? And the key challenge here is that usually there's sort of two ways that we think about it. There's, oops, if my computer will work. There is, you know, phenotype is really disease or healthy, for example, and genotype is what we want to figure out. So traditionally, geneticists think either in terms of forward genetics, right, where we have people that are healthy or sick, and then we try to figure out what mutations underlie this, or usually model organism people will take a genotype and predict the phenotype, right? So this is reverse genetics, this is forward genetics, so you do it either direction and usually do one of them. And the key challenge here is that we do a lot of, well, we as a community, we have to do a lot of work in mice and other model organisms. And then a lot of the sort of direct disease information is coming from human. And so, for example, someone takes a mutation in human that seems to be associated with autism, goes to mouse, spends 3 years trying to make a mouse. That mouse doesn't seem to have any social challenges. So what does that mean, right? Was it wrong? Was it, right? Like, so just to make it extreme, right? Or you go to mouse and you spend years doing screens in mice or yeast or worm, and then you say, well, isn't this great? I found this gene that might be responsible for DNA damage repair, so maybe it's a cancer gene, and then how can we figure out if it's relevant to human? So we're trying to make this cycle a lot more efficient by essentially trying to identify which areas of model organism biology are conserved to human as part of this process. So essentially, close the cycle on forward and reverse genetics. And this is work, again, of Vicky Yao, who really developed all the computational stuff. And then on the experimental side, it's a great collaboration with Colleen Murphy's lab at Princeton, and Rachel Koletsky did the experiments. She's a great postdoc in Colleen's lab. So this was just came out in Nature Biotechnology. All right, so we sort of have 4 parts to this framework that Vicky built. So this is actually a real, you know, for those of you systems people, this is literally a system that biologists can use to do this. First part is what I talked about, right? Building this tissue-specific functional network, except for we build it in the model organism, right? So this reflects the biology of that mouse, or in this case, we're going to show you experiments in worms, so of the worms, in different tissues. Then we're going to make predictions of disease-associated genes by taking examples of genes that have been implicated in this disease, let's say Parkinson's or Alzheimer's, in the human quantitative genetic studies, right, where they looked at human patients with Alzheimer's and without, and they said, well, this gene sort of looks like it's more often mutated in patients with Alzheimer's versus healthy individuals. So what we're going to do is we're going to take the human gold standard, right, for those of you computer scientists, right? So we're going to take the hooks from the human, but we use the model organism network for the relevant tissue to make predictions. We actually automatically choose the relevant tissue, but I'm not going to have time unless you want me to talk about it. And that means that we're going to make candidate disease genes that are specifically biased towards only the areas of biology where there's conservation. Because imagine, if the worm is completely, or the mouse is completely irrelevant to autism biology, then we won't get any significant predictions from the human genes, right? So we automatically are going to exclude the areas of biology that are not conserved. And furthermore, it will not just pick the diseases that are conserved, but even the subparts of the area of where that, right, the diseases are usually, these diseases are multigenic, right? So there's multiple pathways affected. This will focus just to the pathways that are actually relevant to study in that model organism. And so then you can actually use these genes to run the experimental assays in model organisms, right? That's really the power of model organism biology, that we can do experiments very effectively and quickly and be able to now get relevance, at least experimentally confirmed, of genes potentially involved in human diseases that have been experimentally confirmed to be doing mechanistically what you think they're doing, affecting a specific phenotype, and in areas that are conserved on the molecular level between the model organism and human. So does it work? So Vicky made predictions for 25 different diseases with this framework Disease Quest across many different areas. And you can see that it's actually quite robust for diseases that are in diverse areas like cancer, cardiovascular and muscular system, nervous system, and metabolic and autoimmune diseases. And we focused on Parkinson's disease for the experimental phenotyping module in worm because we had really clear phenotypes. So Parkinson's disease, the phenotypes you're looking at are actually very challenging to study without this computational method because you have to look for age-related muscular phenotypes, right? So you can't just look at worms that are unhappy right away. You have to really look across age and see that their movement is affected not just early, but specifically with age, right? So that's very expensive, not just in terms of money, but in terms of time. You have to basically look at thousands of videos and classify this across thousands of worms. So our colleagues have done this, and we identified a number of genes in 4 modules related to Parkinson's disease based on these predictions, and then putting them actually back on the brain network, neuronal network. And here are the 4 modules, and they actually make a lot of sense. There's basically the muscle module, the synaptic, the neuronal module, development module, and metabolic module. And that makes actually a lot of sense in the context of Parkinson's disease. And just to show you, this tends to be all the computer scientists' favorite slide. What this actually means is, so these worms are all perfectly happy in the beginning on day 1. I just didn't have time to show you those pictures. And look at day 8, worms that are totally normal, just old. Like they're moving, they're not moving super fast, but they're just moving. And look now on the left side, and here's the worms that have a gene knockdown that was our very top predicted and confirmed gene, BCAT1. So now, watch do you see how they get stuck in these circles and then they can't get unstuck? So that's actually very similar to a lot of the muscular phenotypes in humans. And just to give you a sense of the scale and why you couldn't do this genome-wide and why you need the predictions is it took about almost 2,000 videos, over 13,000 worms to be able to get statistically significant evaluations for these things. So this is a lot of human power and worm power, I guess. So if we look we've measured a huge number of different features, but if we look, one good measurement is basically how well they can stretch and fold. And here you can see all of our top genes that we tested. Here's BCAT1. You can see that it's a huge outlier, but a number of them are significant. And the cool thing is all the ones the red ones are all the ones that were our gold standard, so positive controls, they are significant. And furthermore, luckily, 23andMe actually published a GWAS study on Parkinson's after we started this experiment. So we actually had a human confirmation of one gene that we predicted and confirmed in worms being completely independently coming out in 23andMe. So this is, I know it sounds like one gene, but these GWAS studies usually find 1 or 2, 3 genes. The fact that one of them overlapped we were incredibly excited. Oops, sorry. Now we have to watch worms again. All right. So furthermore, why are we so excited about this BCAT1? So our collaborators are actually following up on it quite a bit because it looks like BCAT1 knockdown actually exacerbates the effect of alpha-synuclein and actually by itself really causes basically neurons to get unhealthy and die. So if you, like me, originally don't know what we're looking at, essentially this is neurons that are reasonably okay. This wavy stuff means neurons are basically dying, right? Like it's really bad. And that happens if you take out this protein, BCAT1. And furthermore, if we look at humans, BCAT1 is actually expressed much more significantly and higher in the region specifically that degenerates specifically in Parkinson's disease. And furthermore, if we actually look at a study of gene expression in patients with Parkinson's, passed away patients with Parkinson's disease versus healthy subjects, human BCAT1 actually significantly decreased in that specific Parkinson's relevant region, substantia nigra, of PD patients. Now, one question you might ask is, why didn't we just look there and find BCAT1? So many genes are significantly decreased, and you don't know which ones are causal and which ones are just, following. And so you wouldn't have found it coming from human. There's hundreds of genes. And actually, there's very interesting connections here with bodybuilders, for example, have huge hugely, well, not hugely, but very significant and found in multiple studies increase in Parkinson's disease instances. And actually, this gene is relevant to basically metabolism of a particular type of amino acids, branched-chain amino acids, which is what is found in bodybuilding supplements. So none of this is complete proof, but it's enough that I would not suggest taking. But don't record me and don't tell the supplement people. You decide for yourself if you want to still take those supplements. All right, so to summarize, I told you about a deep learning-based algorithmic framework for predicting the effects of any non-coding mutation in the genome without having ever seen those mutations. And then how we can go further and predict tissue-specific gene expression and mutational effect on that. And then we talked about the general semi-supervised method for building tissue and cell type specific networks, even for tissues and cell types for which there is literally not a single data set in your collection, and how we can use those networks to enable real insight into mechanisms of human disease, and I showed you the example of Alzheimer's. And finally, I talked about Disease Quest that really enables us to fuse the power of model organism experiments with human quantitative genetics to really discover and validate human disease genes. So I think I've reasonably well acknowledged the fact that I don't actually do any of this work. I just run around, and my key marketable skill now is Keynote and maybe some Word, although now, I don't know, maybe clicking, barely. So I think I've acknowledged most people who've done this work, but then others involved are also here in red. And we would love for you guys to check out HumanBase. Disease Quest is online, Selene Library. All of those are linked out of our website, function.princeton.edu. Thank you so much. Should I just wave? Sure, go ahead.
Really cool talk, thank you. Is there any future plan to integrate, especially the network data, with protein-protein interaction networks, or how these things respond to drug perturbations?
So very cool question. So the protein-protein interaction data is actually, I kind of, glazed over it, but it's actually part of the input into the functional network. So that's definitely there. And we are thinking very hard about how to make these functional networks. So right now, these functional networks are actually fully connected weighted graphs, and they don't have any mechanistic sense, right? But we actually have other algorithms that do predict mechanistic type of interactions in a tissue-specific way. And unfortunately, right now, the data is so limited that we find that while they look cute and we can write a cool paper about them, in terms of biological discoveries, these functional networks tend to give us more so far. I think as more data comes out, especially as more gold standards come out, that will be better. We are starting to think about how to use that for drug target prediction, sort of in a complementary way, to structure-based methods. So essentially, sort of predict, the way to think about it is really predict the pathways and processes that are hit by the drug, and maybe starting to think about dynamics. So I know Vicky has some ideas, for example, of how she wants to look at dynamics with these networks as well. Are you going to call?
In the back.
Yeah, great talk. For the DeepSea model, you mentioned you validated some GWAS variants experimentally. Is that a CRISPR screen or?
No, so these are basically putting in external luciferase expression assays with essentially just a 200 nucleotide chunk with that, basically that variant, I think that was in autism, right? So that was the proband variant versus the sibling variant.
Great. So, yeah, this was a great talk. So a lot of genetically determined diseases are diseases that we think about as genetically determined. Even the same allele can have diverse phenotypes in different individuals. So 2 questions. Have you thought about how to reverse engineer a suppressor screen out of this, number 1? And number 2, how do you bring environmental variables into this, again, with an eye to something that could be a prevention or a modification?
Right. So really good question. So aside from looking specifically at suppressors, or at least at gene expression controls in either direction, the main thing that we've sort of thought about going towards the suppressor screen type idea, which is many steps before, is to essentially try to combine the GWAS question and the, the impact of variance question with the networks, right? So basically, if you map the non-coding, at least the non-coding variance that you can map to a gene, and then looking at areas of the network that are specifically enriched, right? So it's not exactly getting at your suppressor question, but trying to essentially find at least genes, as a proxy that may be significantly important, or areas of the network that are significantly important that may be integrating allelic signal from multiple different alleles, right? So it's not exactly what you're saying, but it's trying to get a similar question. And we actually have a method that's in part of that autism paper that literally in the network propagates these proband sibling significance scores and looks for genes or sets of genes in the network that not themselves are significant, but have significant weighted connectivity, right? So that basically you get significance out of their neighborhood. So it's not, it's not directly going to find just suppressors, but it gets at this idea of the fact that, of course, it's going to be a network or group or, multiple basically genetic background is going to have a huge effect on your specific allele. So trying to get at the allele context in some way. And I forgot your second question, I'm sorry.
Environment.
Oh, environment, yes.
Phenotype.
Yes, actually I think it's critical. I've sort of started thinking about it, but I think that's actually what Vicky is most excited about. So if she decides to do it, then I'm going to stay out of it. But I know she has ideas, so you guys should listen. No, I think this is obviously, and now there's data basically that's starting to come out on the environment. She has actually a paper that I didn't talk about at all that we're pretty close to writing, looking at splicing in the context of different environments and their effect on Alzheimer's disease. So both environments, basically, this is a mouse study where they have, I think 20 different environmental factors of both physical and mental simulation and then genomic data. So she actually has some results that are very intriguing for how you can start combining that. So of course that comes from, and then I think there's some ideas that she has on looking at wearable devices and trying to bring that. That is going to be really, right, like bringing that together with genomic information is going to be a very challenging question. So we'll see how it goes. But, I think there's some very promising directions at least. We have to think about it.
Any more questions?
I plead a little bit of ignorance in that I'm not obviously an expert on any of this, but I was curious about, I know in certain fields when they have limited sets of data, which you obviously have a problem with, especially with brain tissue collection, is there any way you can sort of fuzz the data and then generate more on that kind of level, or has that been validated to not be effective?
There's definitely subsampling methods, and of course there's, in the deep learning field, there's all sorts of interesting questions of using essentially filters and trying to get at questions that way, and that is effective for very specific types of questions. But it hasn't been, subsampling and semi-supervised type of questions, especially, so in semi-supervised questions, of course, we're more getting at, not quite fuzzing, but, trying to get at the limit of prior knowledge, which in a lot of ways, while it's very hard to get at the specific brain tissues and cell types, although more and more very interesting data is coming out, especially actually the most exciting data sets are actually coming out from, actually I don't know, down the road, up the road, somewhere around here, Allen, wherever the Allen Institute is physically here, they're generating some really cool data sets. I think the most challenge actually comes from the fact that we don't know hardly anything about these specific neurons and cell types, in non-postmortem tissue in human. So I think a lot of it is actually trying to get at the gold standard and trying, that's why I think semi-supervised approaches, learning approaches that can deal with very limited gold standards, and this doesn't always work, but in deep learning, in the genomics case, deep learning has been very productive, right? Or approaches that are truly unsupervised, that don't use prior knowledge. Those sort of things have become more and more relevant.
Good. So I think remember, don't ride motorbikes, don't take unnecessary food supplements, instead walk over to the reception and get on.